So I think we'll get started. So it's my great honor to introduce Professor Donna Ferriero. Donna got her bachelor degree from Rutgers Universities, and she then got her medical degree at UCSF. After she got her medical degree, she also did a postdoc at UCSF and subsequently become an assistant professor there since 1987. So over the years, Donna has achieved a lot of things, and so I will just very briefly mention a few ones. So she is the member of the biomedical science graduate programs. So for those of you who might join Donna for lunch tomorrow, and if you are interested in going to UCSF, so I would definitely suggest that you talk to Donna and try to understand the school program. So she was also the vice dean of academic affairs at UCSF School of Medicine, and currently she serves as the chair of the Department of Pediatrics at UCSF, and as well as the physician-in-chief for the UCSF Benioff Children's Hospital. So Donna won many different awards. So I won't go into details, but to let you know that Donna is a recipient of, she is a member of the Institute of Medicine of the National Academy of Science, and she also served on the National Advisory Council at the National Institute of Neurological Diseases and Stroke. So and she was elected to the Association of American Physicians, as well as the American Academy of Art and Sciences just last year. So without further ado, please join me to invite Donna to give us an IAS distinguished lecture.
Thank you, Tom. I want to thank the IAS for sponsoring this lecture and this wonderful journey, to thank Professor Ip and Professor Chung for the invitation and a great visit. So I want to take you on a little bit of a journey today about why we're interested in repairing the newborn brain. And in order to do that, I'd like to tell you a little bit about what we're faced with in terms of trying to repair the brain. So in the newborn, if you deprive the newborn brain of blood supply and oxygen, you will severely damage the brain, such that you will see by MRI damage to the deep gray nuclei of the brain. You'll see multiple areas of hemorrhage throughout the brain, cortex and deep nuclei, as you can see in the histological photomicrograph in the thalamus here. And this is for Dr. Harrop, a little picture of the cerebellum also showing disease. If the baby is born prematurely, instead of injuring all those deep gray nuclei, the selectively vulnerable regions tend to be in the white matter. So it's the oligodendrocytes that are damaged, and these lead to these cyst-like formations in the periventricular white matter and damage throughout the brain. The good news is, even though you can damage or injure the newborn brain, and here on this diffusion-weighted MRI you see damage in the thalamus and caudate putamen, with elevation on spectroscopy of lactate suggesting oxidative stress occurring in the brain. There is a prolonged period of time in which this brain is undergoing continued injury and potentially a chance to repair the brain. So 15 days later, we see extension of the injury on this MR image into different regions of the brain. And now the oxidative stress is diminishing, but so is the neuronal integrity. NAA is N-acetyl aspartate, marks neuronal integrity, At 16 hours, we see a normal peak, but by 15 days, we know that there's been substantial brain damage. So is there anything we can do about this? We've looked at many ways of manipulating this pathway, and the pathway goes on for days and weeks, and I'll show you that time frame in a minute. But once you prohibit the brain from receiving blood flow, you end up with energy failure, you cause excitotoxicity, and that results in both apoptotic and necrotic cell death, energy requiring and explosive cell death, as well as delayed cell death. These processes all set up an inflammatory response, and it depends on how you manage these early events, whether you can repair, restore, and promote plasticity and recovery in the brain. The good news is for the newborn brain, we can see that there's a prolonged period of time. Look at this scale is in weeks. The repair isn't happening right away. So we have time. If we can blunt these processes like oxidative stress and excitotoxicity, we could potentially lessen the inflammation, cell death, and promote repair. And that's the goal of current therapies. And right now, in the United States, in Europe, Australia, and China, we are cooling babies' brains by cooling the whole baby down to 33.5 for 72 hours. In doing that, we reduce these early phases of damage in the hopes that we can then get in later to repair the brain. We haven't quite figured out that part yet, and that's what I want to talk to you about in this lecture. And this is just an example to show you by MRI scanning. This is a human newborn at birth who sustained a stroke. And for those of you that aren't familiar with stroke, everybody thinks about stroke as a disease of the elderly. Well, it turns out that newborn babies have stroke as frequently as an elderly person. So it's really quite a common problem that we need to deal with. At 3 months, we're starting to see lessening of the damage, but we get degeneration of the tracts that are originating in this area of the middle cerebral artery. In babies who have severe hemiplegia, we can look using diffusion tractography and show that there's diminution of those tracts that has occurred over time. Whereas in children with mild hemiplegia, we know the stroke wasn't so bad. So what we want to do is get to that point. Even if there's damage, try to promote repair and healing. So one of the ways that we've tried to think about this, at least in animal models, is to look at hypoxia-inducible factors, because these are the earliest responders to the injury. And HIF is induced by hypoxia and stabilized by a number of factors that then allows for transcriptional regulation of a number of genes that help the brain to survive. And those include things like preserving energy through glycolytic enzyme and by the promotion and release of growth factors like VEGF and Epo. And this is these are the factors I'm going to talk about, mostly erythropoietin, to show you why we think this could be a potential cure. And this is not so far-fetched, because at the end of this talk, I'm going to tell you about some clinical trials that are ongoing right now with Epo in the newborn brain that are quite promising. So how do we know this even matters in these animal models? So if we create a stroke in a newborn rat or mouse, and I'll show you how we do that in a little while, we can look for a hypoxia-inducible factor in the brain, and we see it comes up quite quickly in the area, the ipsilateral side, this is the contralateral side, as early as 4 hours after the occlusion of that artery. And the good news is that HIF is coming up in neurons where we want it to. We want it to induce the preservation enzymes, like the glycolytic enzymes and the growth factors in neurons. And we don't see HIF coming up in astrocytes, which is a good thing. GFAP is a marker for astrocytes, and astrocytes are cells that create scar in the brain. So we want to diminish astrocytes and promote newborn neurons. We also know, by virtue of creating some hypoxia-inducible factor deficient mice, that hypoxia-inducible factor is important for the repair and recovery and the injury process, because if we knock out HIF in neurons, we can show that these mice actually have increased injury in the brain, and specifically in the cortex, where most of the damage lies. So to remind you again about the signaling pathways, hypoxia is inducing hypoxia-inducible factor 1 alpha and stabilizing it, thus allowing for the transcription of the erythropoietin gene. Epo receptor is also coming up, and the good news is that both Epo and Epo receptor are expressed throughout the brain in multiple cell types. The question is whether exogenous Epo is needed, or is there enough endogenous Epo to allow for repair and recovery. So we know that Epo is made in neurons and astrocytes, and we know that these cells talk to each other via their receptors. There are also Epo receptors on microglia, which are the inflammatory mediators in the brain, on the oligodendrocytes that are responsible for myelinating axons and preserving that white matter that we see damaged in the premature baby, and also on endothelial cells, so for preservation of blood-brain barrier integrity. So what about the HIF target gene Epo? Is it upregulated after this newborn stroke? When we look, we can stabilize HIF using desferoxamine, and we can see that it is upregulated in a similar time frame as HIF. HIF came up at 4 to 8 hours. Epo is coming up a little bit after that. And at 8 hours, Epo, we can see when we double stain that the Epo is expressed in neurons. However, if we look 1 week later, we still see Epo, but this time Epo has switched to a different cell population where it's being expressed, and it's now being expressed in astrocytes. We don't want that to happen. So what can we do to manipulate this process? This is possibly where we will need to look at exogenous Epo. But we needed to learn a little bit more about the signaling of Epo in the brain, and so we looked at some downstream signaling molecules, and we looked in particular at ERK and at PI3 kinase. And we found that ERK signaling was definitely increased. So let me take you through this a little bit. The VO stands for the animals who have had the stroke but didn't receive Epo. EO are the animals that had the stroke but received Epo, and this is at 8 hours and 32 hours after the insult. We see no change in total ERK, but we do see an upregulation of ERK, phosphorylated ERK, in the deep gray nuclei, the caudate, in the Epo-treated animals at 8 hours, which diminishes over time, and then we see it in the cortex and hippocampus as well, all happening early. So we know that Epo is signaling and activating in the setting of hypoxia ischemia. And looking at other downstream regulators like PI3 kinase and AKT1, we can also see in this situation, we're just looking at cortical neurons in a dish. We use oxygen and glucose deprivation to equate to hypoxia ischemia in vivo. And we can see here that although there's no change in total AKT, we see that upregulation of phospho-AKT early at 4 hours. And when we look in that system, if we inhibit the AKT with phosphorylated AKT with warp mannan, we can blunt it in these cortical neurons in culture, and here you can see that they're pretty much wiped out in the presence of wortmannin. We also know that Epo can affect and interact, as I've just said, with AKT. And here we're looking at a different in vitro model. It's a hippocampal slice culture. In this hippocampal slice culture, we're looking at depriving also that culture of blood supply. So it doesn't really have a blood supply, so we use OGD again, oxygen glucose deprivation. And here we're looking to see the effect of Epo on the neurons in that hippocampal slice using a marker for immature or migrating neurons called double cortin. In the cultures that aren't treated, we don't see much expression compared to control. But in the Epo treated, they're looking just like the controls, despite the fact they've been exposed to the OGD. If we use an antibody to the Epo receptor, we can obliterate that response, so we think it's effective. And here again, inhibiting AKT shows a similar negative response. ERK is also involved in VEGF signaling, which is the other growth factor that we think is very important in repair in the brain, probably more so in terms of angiogenesis, which I'm not going to talk about today. But here we can see, if we look at HIF and VEGF after injury, these are now postnatal day 10 rats that have been exposed to hypoxia ischemia. We occlude one of the arteries, we put them in a jar that creates a global hypoxic environment, and they end up infarcting that hemisphere. And we can see that, again, this similar time frame of upregulation of HIF and VEGF, ERK, phosphorylated ERK with no change in total ERK, and inhibition using an ERK inhibitor. And we know that VEGF is upregulated in neurons after the neonatal stroke in that model that I'll describe in more detail in a minute. And here again, we see nicely that VEGF is coming up in neurons, and I hope you can appreciate the yellow cells here where we want it to be, and it's also co-localizing with HIF, as we can see here. And this is at 8 hours in the cortex. So there are other cell types, and I don't want to forget about them. And we think there's a lot of crosstalk between, and paracrine signaling, if you will, between the neurons and the astrocytes in the brain in the setting of hypoxia ischemia. Now, this is a situation where we're just mimicking oxidative stress by exposing cells to hydrogen peroxide and looking at the effect of Epo in that setting. And if we look at the astrocytes, we can see that Epo can protect them against this oxidative stress. 500 micromolar, which is quite high, of hydrogen peroxide will kill the astrocytes. This is cell death here. But if we give Epo, we can bring it pretty much down almost to control levels. But Epo will not protect the microglia from oxidative stress in this situation. So inflammation then can continue even in the setting of oxidative stress. Now, astrocytes do serve a purpose, especially early, because they can dampen the excitotoxic response by taking up glutamate. So we want them around early, but we don't want them to over-proliferate. So let's focus a little bit more on Epo. Just to remind you, it's a 34-kD glycoprotein that is really a cytokine, and it's quite pleiotropic. It has effects on every single system. And of course, we're interested in the CNS, but it affects all the systems that interact with the CNS. And we're most interested in what Epo is going to do in terms of proliferation. There have been numerous studies, we're not the only people obviously studying Epo and neuroprotection, and in numerous preclinical studies of whole animals and cell culture studies, all of them suggest, both in the immature animal and the mature animal, that EPO is neuroprotective in this setting of ischemic injury. However, there's only a modest response, so maybe half of the brain injury is reduced in this setting. So there's more to be learned and gained from trying to pick at this a little bit better. So we were trying to figure out how could we end up looking at this time frame in the newborn animal model. So this is what we do. We meaning a very talented neurosurgeon in my lab. We create focal ischemia and then allow the brain to reperfuse itself by inserting a nylon siliconized suture up the common carotid into the internal carotid, and we stop at the bifurcation of the middle cerebral artery. We hold it there for a certain period of time that we know will create an infarction. And here's the full protocol to show you what the diffusion-weighted image looks like. So we only take those animals once we've stroked them and look at the animals that have diffusion-weighted changes by MRI. So if they show this injury, then we'll then segregate them and give them erythropoietin immediately after reperfusion, 24 hours, and then 7 days later. We will then label cells with BRDU to see which cells are being born, and we do testing, sensory motor testing at 2 weeks and at 3 months, and we also do volumetric analyses, immunolabeling, and confocal analyses to see which cells we're left with at 6 weeks. We use 4 groups of animals because we need to show that sham surgery doesn't do anything in these animals. We also give the shams EPO, but the real groups we're interested in are the VO and EO, which are the animals who have been stroked, those that have received no treatment or vehicle, saline, and those that have received EPO. And the good news is that EPO does improve both structure and function in these animals. If you look at 2 weeks with just 1 dose, you end up seeing a beneficial effect. But if you wait 3 months, you've totally lost that effect after only 1 dose. So we have to give those repeated doses, and it's actually the last dose that we think is the most important dose, that 7-day dose. And if you remember, I showed you earlier that EPO was switching its location, or its expression, if you will, in the brain at 7 days after the insult from the neurons to the astrocytes. So we need to give exogenous EPO to block that response. And in that situation, we can show pretty much complete or almost complete preservation or repair of the brain. So we don't quite know yet whether we've just completely eliminated that cascade of events from occurring or if we've actually caused new cells to develop and populate the brain. And likewise, we've been able to show functional repair using the Mars water maze where we allow the rats to swim around, and the EPO-treated rats will perform exactly the same as the normal rats. So we want to get at this question of neurogenesis, and there's a big difference between embryonic, obviously, and adult neurogenesis, where adult neurogenesis is primarily situated in the subventricular zone and hippocampus in the dentate. In the newborn brain, there's a much bigger ventricular zone and subventricular zone with tangential as well as radial migration of newborn cells. So the cells that we're interested in looking at are these neural stem cells that will go on to become neurons to populate the brain, oligodendrocytes to myelinate the axons of those neurons, the 2 very important cells, and astrocytes, which are important because astrocytes are kind of the cleanup machinery, if you will, of the brain. But if there is too much of a response, a too much robust response of astrocytic proliferation, you end up with a scar. So we looked at what was happening in the brain after stroke. This is an adult where we looked at the subventricular zone here outlined in white, and we stained for double cortin, which is that marker of immigrating immature neurons. And we could actually see those cells, and here in this box blown up, you can see a chain of cells migrating actually to the infarct site. And when we stained that infarct site, we saw those cells, and then we stained for neurons, DAP32, which are the intrinsic population. They became the cells that we wanted them to become, but there were very, very few of them, suggesting we needed to do something exogenously in this situation. In the newborn brain, there's much more chaotic distribution of newborn cells migrating tangentially and radially. And surprisingly, we see this robust expansion of the subventricular zone rostrally and throughout the subventricular zone. We thought that was good, but it turns out that actually SVZ overexpansion actually correlated with more injury rather than less injury, as if the cells weren't able to get out, develop, migrate to the appropriate source. And here you could see, in terms of SVZ population, that Epo actually decreased that SVZ expansion. So it was a little counterintuitive, but decreased overexpansion resulted in reduced infarct volume. So what's going on? Well, we thought, well, maybe Epo is increasing neurogenesis after injury. So we looked both in vivo and saw, yes, in those Epo-treated animals that had stroke compared to the vehicle-treated animals, that there was an increase in newly born cells that became mature neurons. And when we looked in the dish as well, and this is in a hippocampal progenitor cell culture, and looked at newly born neurons marked by 2G1, we could also see in the presence of hypoxia and Epo an increased number of those cells. And interestingly, even in the setting of normoxia, Epo had a beneficial effect. So we wanted to look a little bit more carefully at this, so we labeled these stem cells in the subventricular zone with a GFP-expressing lentivirus right after birth at P1 by direct injection. We created the stroke at day 7. We gave the 3 doses of Epo after injury 24 hours later in 7 days. And then a week later, 3 days later, and 2 weeks later, we looked for double labeled cells of the GFP and double cortin, the immature marker. So what we saw was that Epo did shift cell fate of these SVZ stem cells towards neurogenesis, just what we wanted. So when we looked at the GFP positive and double cortin positive cells in the striatum, and that is what we're looking at here, we see in the red the Epo-treated stroked animals marked expansion, marked proliferation at P10, and a sustained effect at P21. And even in the normal animals, the sham animals treated with Epo, we saw increased cells. So maybe that's why Lance Armstrong was taking Epo. I don't know, he wanted more neurons so he could win the race. So we also looked at, just to make sure that GFP was in the right place, and here it is in the striatum, where here's the SVZ right there, lateral ventricle, here's the cortex. This is the deep gray nuclei, the striatum, with lots of GFP positive cells that have migrated to the infarcted area in the ipsilateral striatum. in the Epo-treated animals. If we looked in the vehicle-treated animals, we didn't see in the stroked animals any cells in those areas. And if we look at P10, we can see GFP-positive cells. At P21, again. And if we look at the proliferating cells, we see the marked proliferation in the Epo-treated group. We could also look for the migrating neurons, the double cortin neurons like I showed you earlier, but this time they are labeled now instead of with BRDU. Right from birth, they're labeled with the GFP lentivirus. And again, we see the same thing at P10 and P21 in the ipsilateral striatum, this proliferation with exogenous EPO in the stroked animals. And those cells do become mature neurons, as we can see here with GFP new N. DAPI is just the blue staining the nuclei. And here we see, again, the EPO response. So we wondered if it was just neurogenesis or if we were also stimulating that other cell type I told you about, the oligos, and what was happening with the astrocytes too. Remember, the oligos are the cells that are myelinating the white matter and allowing those axons to stay healthy. The astrocytes are the cells creating scar. So when we looked for increased oligo precursors, we did find them staining with O4 and GFP. We again saw them in EPO at P10, at P21. And when we looked at GFP, we were very relieved to see that there were much fewer astrocytes in, at P10 and also sustained at P21. When we looked at hippocampal progenitor cells in the dish specifically, again, I showed you this slide before looking at the 2G1 positive cells, showing you that we were getting new neurons with EPO, but also what we're getting is less astrocytes and less scar tissue. And here's no EPO, showing you these very fibrillar astrocytes here in green, and in the EPO-treated cultures, essentially none. And Laura Jancy just recently showed a similar effect using oligodendrospheres after transient hypoxia ischemia and using a marker for early oligodendrocyte progenitors, PDGF-alpha. She saw an increase in the presence of recombinant EPO. So our cell fate studies seem to suggest that EPO treatment after stroke does result in newly born cells by Ki-67, a production of immature neurons that also become mature, a favoring of oligodendrocytes over a situation of astrogliosis as measured by GFAP. And after transient hypoxia ischemia in vitro, Laura showed this in OBC progenitors. So does it matter? So we have a bunch more cells. Are we going to be smarter? Are we filling in the holes? So we looked at synapse formation initially by markers, and now we're looking electrophysiologically. I don't have that data to show you today, but I can show you our results with synapsin 1 after the MCAO. And if we look at total synapsin expression, we're not really seeing much of a difference in the EPO-treated animals. However, if we look at phosphorylated synapsin in the cortex at P13 and P17, we see that at an increase over time in the phosphorylated synapsin, as well as immunofluorescently positive puncti at P17. If we look at the other side of the synapse and look at postsynaptic density 95 expression, we can see similar things happening with EPO increasing at P17 and a trend toward increase in the puncti. So then we were trying to figure out, well, how can we give the EPO? We can probably just give it peripherally, hope it crosses the blood-brain barrier, and in effect, I'm going to tell you clinically, that's what we're doing, and it seems to be working. But we were also looking at delivering stem cells to the animals to see if that would be a good way to deliver growth factors. So in collaboration with Kobe Heinian from Utrecht and her postdoc, who is now working in my husband's lab, Cindy van Veldhoven, we looked at mesenchymal stem cells. And we engineered them to produce BDNF because we knew brain-derived neurotrophic factor had been shown previously to promote plasticity and recovery in adult models. So using those cells, we were very delighted to see that when we looked at volume loss, here are the sham animals, and here's the vehicle-treated animals showing a tremendous amount of volume loss, the mesenchymal stem cells with a little bit, with less, and the BDNF engineered cells showing even better response. We gave the stem cells intranasally. They do cross the cribriform plate, and they do populate, but they're not sustained over time, and I'll speak to that in another minute. This is just showing neuronal changes, and this is showing white matter changes by MBP, showing beneficial effect of the stem cells. So that's good. Here again, I'm showing you form, but what about function? So if we look at functional recovery in these animals, and this test is called a four-paw initiation test. And what happens is you put the animal, the rat or the mouse, into a cylinder, and normally it will rear with both hands going up against the jar. If the animal has a stroke and a hemiparesis, it will not raise that forepaw and will show a forepaw preference. So the good animals are down here, showing no forepaw preference. The bad animals are up here, showing complete forepaw preference. And the mesenchymal stem cell treated animals here and here with BDNF early showed a very remarkable response at 14 days, 1 week after the injury. But when we waited a while, they kind of lost the effect, and it looked the same as just the mesenchymal stem cells as the same. So we don't know whether it's a dose effect or not. But clearly, and here's another test. This is the adhesive removal test. The animals will stick on to adhesive tape, and if they're injured, they have a prolonged period of time or increased latency. They can't get that paw up, and that's what you see here. But the mesenchymal treated animals, as well as the BDNF mesenchymal treated, did well. When we looked for cell proliferation in the subventricular zone, a month later after the stroke, we saw that there was cell proliferation in those regions, and I'll show you a photomicrograph, but this tells the whole story. It's not an overexpansion, but there were EDU, BRDU positive cells in both the mesenchymal stem cell and BDNF mesenchymal stem cell populations. And we did see the Ki-67 proliferating cells in the subventricular zone at that time. So it's a prolonged response after a single sniff, if you will, of mesenchymal stem cells. And this is just a picture. I just wanted to show you the proliferating cells in that subventricular zone. Here you can see the BDNF population and the mesenchymal cell population. So what we think's happening is that the stem cells are creating a favorable microenvironment, but then they're dying. So they're releasing growth factors. So it's another way of giving growth factors. So it's a good way to give growth factor. It may be easier in the nursery to inject stem cells into the nose of a newborn rather than go for infusions of a protein, which can cause serious problems. But we think it's those growth factors that are allowing for proliferation of these neural progenitor cells into the appropriate subtypes that I showed you happen over time. So you can say, great, rats doing well after a stroke, cells in a dish, they look pretty good. What about humans? So we have been doing clinical trials, even in with good preclinical support for Epo. And there's actually new primate data from Sunny Jewell's lab at the University of Washington showing that baby primates whose umbilical cords were clamped, when mom was given the Epo, the babies did not develop cerebral palsy. There were 2 small human trials, one in China and one in Egypt, with a few patients, not big studies, but they were suggestive of safety and efficacy up to 6 months after the administration of Epo. We've done a phase 1 trial of Epo in newborns who have had hypoxia ischemia. It's safe, and the Epo gets from the blood into the brain where we want it in doses that are comparable to those that we see efficacy in the animal model. So we know that we're in a good optimal dose range. There are a number of ongoing studies. There's a phase 3 trial in Paris that's nearing completion. Now all of these studies are being done in the setting of hypothermia. Remember I told you that we cool these babies after hypoxia ischemia to blunt those early metabolic responses of oxidative stress and excitotoxicity. So the Epo is being given after the hypothermia or in conjunction with the hypothermia, and that's what HT is. Mariana Basurga is in Utah. She's done a phase 2 with a Epo derivative that looks promising. This is Yvonne Wu's study of Epo, now just completed a phase 2 study, and her early MRI data show benefit in the Epo-treated children. And she has now put in an application to the NIH to do a full-fledged phase 3 trial. And then in preterm babies, remember I told you it's the white matter that's important when babies are deprived of blood in the early gestational age groups. And this study out of Switzerland shows that with secondary outcomes, that high-dose Epo within 42 hours after birth was associated with reduced risk of brain injury on MRI. So it's a short-term outcome, but at least it's a positive outcome showing less injury in the Epo group compared to the placebo group with a pretty good risk ratio. So just to put it in perspective, there are a number of trials of hypothermia that suggest that we need only 7 babies to treat to get benefit in one, which is a great number to treat ratio. And all of the studies, whether you cool the head itself or cool the whole body, show benefit, but the benefit is small. So we're only getting benefit in maybe 30 to 40% of the children. So I think what that suggests is that even though we blunt those early phases of injury that I showed you, the oxidative stress and the excitotoxicity, it's not enough, that we need to do something that will modulate inflammation, which we haven't gotten to yet, but maybe melatonin is something that we could use there, and that is also in clinical trial, and also modulate repair, which is what we're hoping to do. And in the phase 3 trial is designed to give Epo 24 hours later up through discharge of the baby from the hospital. So we're hoping that we can reduce major neurodevelopmental disabilities in survivors. And with the idea that we're not just improving how the brain looks, but how the brain acts. So I just want to finish by thanking all the funding agencies that have helped me: Cerebral Palsy, American Heart, March of Dimes, the NIH, and private foundations, and a whole host of folks who have contributed to some of the work that I've showed you here today. Thank you for your attention.
Thank you, Donna, for a very interesting talk. So now the floor is open for questions.
Just curious, do you think that the Epo would be possibly effective for adult stroke patients?
Actually, there was a clinical trial in Germany given for adult stroke. The problem in that trial was it was stopped early because the adults got TPA early, as is standard therapy for adults with stroke, to break up the clot. And in the adults who got TPA plus Epo, there was an increased mortality. So the trial was stopped, and people are afraid to go there because they don't want to recommend not using TPA. They think it's the TPA. The folks that did die died from hemorrhage. Yeah.
Donna, you mentioned that you need to deliver 3 doses for the animal models. One right after, and then 24 hours, and then 7 days.
7 days.
7 days. And you mentioned that the one at 7 days is critical.
Yes.
So I'm just wondering, for these 3 doses, I mean, do they all act on neurogenesis, or do you think they have a different effect, and together, it brings about the positive outcome?
Yeah, I think it's a great question. In fact, somebody else asked me if we should just give it at 7 days and see if that alone is what we think. We think that 7-day dose is modulating cell proliferation and affecting repair. Epo is a cytokine. It is an antioxidant. So there have been plenty of studies to show reduced oxidative stress. We now have MR evidence using spectroscopy, and we do see less oxidative stress in the kids after those early Epo doses. So it is working. Whether it's critical that it's working at all of those phases, we don't really know. But the way the studies have progressed, nobody is willing to give up those early dosing because they're afraid that, they have to hit them hard early and then continue. So, but we don't know. It's a great question. I'm sorry. Yeah.
Actually, I'm a little bit confused about one question that, as you mentioned, 1 week after the stroke, the Epo receptor on the astrocyte was increased. So I wonder, are they turning the astrocyte to new neurons, or are they melting the scars to, as you said, after they formed enough neurotrapping factors are killing the astrocyte?
Yeah.
Which part?
Yeah.
It's a great question. I don't think that Epo is as active on astrocyte proliferation as it is on neuron proliferation. So it seems that the neurons are winning over the astrocytes. It's not killing astrocytes. And in fact, as I showed, if you look at Epo in a dish of cultured astrocytes, it will protect astrocytes from oxidative stress. So I don't think it melts scar, kills astrocytes. I just think it shifts cell fate toward a more neurogenic pathway.
So are you talking that they will make the astrocyte into new neurons?
No, I think it's the neural stem cell that's becoming a neuron, not the astrocyte.
Thank
you, Professor, for your nice talk. I have two questions. The first one is wondering if you delivered the lentivirus GFP into P1 mice, and then you found that even in the low-stroke model, the Epo treatment will increase the neurogenesis at P21, but I didn't see the increase in the P10 in the Epo-treated but non-stroke model. There is increase in both, only in P21 but not in P10. But in the stroke model with Epo treatment, there is both increase in P10 and P21. So I'm just wondering what happened here.
Well, I think, I don't expect you to remember this slide, but if you look at proliferating cells, there are more proliferating cells by GFP in the injured areas. So you need the injury to get a more robust response. Epo will still increase cells, but it does it much more effectively, if you will, if the brain is injured.
Thank you. And the second question is just wondering whether there is a different localization of Epo in regarding whether it is endogenous or the extradogenous. Does endogenous Epo localizing neurons more, and then exogenous localizing the GFP positive astrocyte?
Yeah, I don't think I can answer that question. I think Epo is throughout the brain. There are certain areas of the brain that have a higher expression of Epo-positive neurons than others. Those tend to be the areas where neurons are more metabolically active. And in terms of whether, what was the second?
I mean, there are some endogenous Epo as well as exogenous. So I'm wondering whether there's a different localization or distribution of the endogenous versus exogenous.
Yeah, I don't think, we haven't tagged the exogenous Epo to find out which cells, where the Epo receptor it's binding to, so I can't answer that question. But thank you for
Epo can promote the genesis of oligodendrocytes.
Yeah.
So I'm wondering, is that because the white matter is more vulnerable to the brain damage, so we need more oligodendrocytes to fix those paths, or it's just because oligodendrocytes is more important for the general brain damage repair?
Yeah, there's a very unique selective vulnerability in the developing brain. So in the very immature brain, it is the oligodendrocyte that is much more vulnerable. And you will cause maturation arrest if the organism is exposed to hypoxia ischemia of the oligodendrocytes. As the brain matures, the selectively vulnerable cells are neurons, not oligos, although oligos are damaged. But you need oligos anyway because the neurons have axons. If you will end up with Wallerian degeneration, axonal demise, if you don't continue to myelinate. So you still need to have a response via the oligodendrocytes.
Another question is, do we have a way to remove or reduce the oxidative stress after brain?
Yeah, that would be great if we did. I think really that's what we've tried to do with hypothermia. Blunt the oxidative stress response by decreasing metabolism, decreasing energy failure, decreasing the production of superoxide and hydrogen peroxide in the brain. But there are other drugs that people have tried and in animal models seem good, like melatonin, which seems to blunt oxidative stress. Vitamin K, vitamin E, all can reduce oxidative stress. There are a whole host of antioxidants, if you will, that seem to be beneficial, at least in rodent models. But in terms of the humans, it seems that the hypothermia seems to be the safest, best bet at the moment, although melatonin may be a good. I showed the cerebellum, but you came in late.
What can I say? There's so much people here, I can't possibly. But it does raise a question, which is the studies in the babies were all done with hypothermia as standard of care. Have you ever tried dropping your incubator temperature to 33 and asking whether OGD in vitro or cooling your rats to see whether any of the biology that you're measuring changes with hypothermia?
Yeah, we've done all the hypothermia studies not in the dish, but in the animal models. We've used mice, rats. We and many other labs have done hypothermia, and the preclinical studies look just like the human. Looks better. Piglets especially, because you can get better assessment of the metabolism. You can measure spectroscopy in a piglet brain, and you can show reduced oxidative stress in a piglet with hypothermia.
Hi, Professor. Thank you for an interesting study. I have a question is that in your animal model study, you said that in the stroke protocol, you have to inject the EPO 3 times because of the role shift of EPO from the neuron to the astrocyte. So I was wondering whether this can be realized in the clinical trial. So how can you do the EPO injection to the infants and try to control the effect of EPO within the neurogenesis? Whether it might contribute to some neuroinflammation aggregation that might lead to some side effect.
Yeah. Well, that's always the balance, right? That's why we first try to do phase 1 trials to show that there's safe and no adverse effect of the therapy. And we hope that the therapy we're giving is doing what we think it's going to do. So the babies are getting dosed at 7 days. But they're also getting dosed at day 1, 3, and 5. So they're continuing to get the EPO along the way.
So I'm just wondering, any information you know about the EPO or hypothermia clinical trials for adult stroke or adult brain injuries? So what those results with neonatal, what would be your
Yeah, it's similar to the question that was asked earlier about EPO for stroke. Doesn't, in that one trial out of Germany, it looked like there was increased mortality, so they stopped the study. Hypothermia is used in adults it works quite well for cardiac arrest. Does not work well for head injury, traumatic brain injury, or spinal cord injury. And it is used for stroke as well. I don't know if there are any ongoing studies but I don't think anybody's going to combine hypothermia and EPO in the adult because of this issue of the mortality with TBA.
So I think the major difference is because of the neuron the way it got protected, or because of show this nice neurogenesis from this young animals?
Yeah. So I think that there is some element of protection, at least in the human, from the hypothermia, and that those cells that possibly would go on to be injured might not be because of the hypothermia. But I think that the EPO is really working at the later phases of the processes of this injury cascade, if you will, and is more focused on the repair end rather than on prevention. So I think it's not preventing injury. I think it's promoting repair. But that's still best guess. Yeah.
So how about can treatment be used for other developmental disease in babies? For other developmental disease in babies?
Yeah, so right now it's only being trialed for those injuries that occur acutely. So hypoxia ischemia from birth asphyxia, or stroke from an occluded vessel. It would not be used, I don't think, in the setting of encephalitis in the newborn, nor would I think it would be used for a genetic cause like a channelopathy that causes severe seizures or brain malformations. Although we will end up finding in our studies that kids who look like they had hypoxia ischemia will get treated with Epo, just because we won't have the brain images until later. And we may end up seeing an effect. There may be an effect in a child, for example, who has Down syndrome, who ends up also being hypoxic ischemic. You know, right now, all of the trials have excluded any child who had a congenital malformation or an infection. So we don't know the answer to those states, and it may turn out by being inclusive in the phase 3 studies, we may see some benefit.
Okay, so if there's no further questions, we have a reception right outside of this auditorium. So if you have further questions, please join us there. And let's join you to thank Professor Ferriero for this.